Hi class, welcome to your biochemistry for medical laboratory science. And for today's lecture, we will be discussing your modules 15 up until your module 17. So for the periodic 2 examination, the coverage will be from modules 10 hanggang module 17. And for today, ito na yung last natin na discussion for period 2. Okay? So again, ang coverage po ng ating examination will be from modules 10 hanggang module 17. But before we dive into our main discussion for today, let me just give you an additional information in regards to last week discussion. So ano-ano po ba yung mga topics na na-discuss natin last week? It is mainly about your urea, your amino acid, and then your protein, and then your ammonia. So let us start with your urea. Urea, its clinical significance includes abnormal serum urea levels may be due to pre-renal, renal, or post-renal disorders. Pakitandaan po na si urea, it is a metabolic waste product. It is a product of your protein catabolism. So it must be excreted in the kidneys through your urine. Okay, so increased serum urea may be due to renal failure, glomerular nephritis, urinary tract obstruction, congestive heart failure, dehydration, and increased protein catabolism. So our urea being a waste product must be excreted. Okay? At hindi po dapat siya nagsistay longer at hindi din dapat mas mataas yung level niya inside our circulatory system. Kasi nga po, it can be alarming. Ibig sabihin, there is something wrong with your function, biological function. Merong pwedeng may problema ka sa iyong organs or on this particular enzyme. Madami pong mga contributing factors. So, decreased serum urea can be due to severe liver disease naman po, vomiting, diarrhea, and malnutrition. Next, there is a term on what we call your blood urea nitrogen or your BUN. It is an older term still in use, and the terminology was based on previous methodology where nitrogen was measured. So si blood urea nitrogen is one of the many NPNs, yung tinatawag natin na non-protein nitrogenous waste, that can help us to detect kung meron pong mga malfunctions or merong something wrong with our enzymes as well as yung ating mga organs nga po. Okay? Kasi nga being the urea as a waste product, it must be excreted. So pag na-detect po siya sa ating liver or sa ating serum, particularly on the blood, ibig sabihin meron po talagang problema. So next naman po si ammonia. Ammonia for the regulation, ammonia is produced from the deamination of your amino acids. Tandaan na ang ating functional group ng inyong amino acid, meron kang R group as your side chain, meron kang carboxyl group, and then on the left side, kung nai-imagine, meron tayong tinatawag na amine group. Okay, the hepatocytes or your liver cells convert your ammonia to urea for excretion. So tandaan na si ammonia is a product of the degradation of the amine group. So parang product din po siya ni protein catabolism. And then your hepatocytes convert ammonia to urea for excretion. Doon na po sila papasok sa urea cycle. Okay? With severe liver cell malfunction, blood levels of ammonia may increase. And then pakitandaan na si ammonia ay neurotoxic siya. So again, ano bang sabi natin? Bakit po neurotoxic si ammonia? Si ammonia kasi, pagka nakarating yan sa ating blood circulation, okay, or into the bloodstream, it can gain proton. So from being an ammonia, it can be an ammonium. And this ammonium may travel into our CNS o yung tinatawag natin na central nervous system, and it can lead to cerebral edema. Okay, bakit po nagkaroon ng cerebral edema? Kasi nga po, nung nag-travel na siya papunta doon sa ating central nervous system, nagkaroon ng increased intracranial pressure. Increased intracranial pressure. Okay, nung nagkaroon ng cerebral edema, it can lead to comatose na po. Okay, and then this comatose is neurotoxic. So ayun po, kung bakit hindi dapat magkaroon ng increased level of the ammonia into our circulation, especially doon sa ating bloodstream. Next, for the type of specimen and storage, kapag ka naman isa-subject na natin siya for testing, especially in the clinical chemistry section, the blood or the specimen to be used must be venous blood, again, venous blood, free from hemolysis. And then it must be placed on ice immediately. Bakit po hindi dapat hemolyze yung samples natin? Kasi kapag na-hemolyze yan, yung mga tinatawag nating mga nutrition or yung mga ibang vitamins or such as yung mga protein na na-degrade galing doon sa inyong RBC, it may lead to false increase or false positive kapag ka-hemolyze yung samples ninyo. Okay? Kasi yung ating RBC, red blood cells, marami yan mga iba't ibang vitamins, and then meron niya mga proteins, meron din niya mga carbohydrates, meron din niya mga lipids, mga ganyan. So dapat po venous blood free of hemolysis and must be placed on ice immediately. So the blood collected in your EDTA, okay, may be used. And then centrifuge the sample within 20 minutes of collection and remove your plasma. And then the plasma stable up to 3.5 hours in ice bath, and then stable several days frozen. So the clinical significance of the ammonia is increased plasma ammonia levels may be seen in hepatic failure or your Reye syndrome. Again, tandaan po, isa po itong recall on your board exam, and then as well as a clinical chemistry, clinical significance of increased plasma ammonia levels may be seen in hepatic failure and Reye syndrome. So ito yung test methodology niya. Hayaan ko na po itong i-discuss sa clinical chemistry ninyo. And then yung mga interferences natin that may occur is it includes incorrect handling of the blood sample and ammonia contamination. So usually dapat ang reference range niya is ganito lang at around 11 to 32. So now let us proceed with your lipids and lipoproteins. Your lipid structure is number 1, the fatty acids exist as short, medium, and long chains of molecules that are major constituents of your triglycerides and phospholipids. Minimal amounts of fatty acids are bound to albumin and then circulate free as unesterified in the plasma. So next, yung triglyceride naman natin, it is the major storage of your fats. Triglyceride is formed from one glycerol molecule with 3 fatty acid. Again, ano yung chemical structure ng inyong triglyceride? It has one glycerol molecule and meron siyang 3 fatty acid molecules attached via ester bonds. Okay? Number 2, triglycerides comprise of 95% of all fats stored in your adipose tissue. Number 3, triglycerides are transported through the body by chylomicrons and very low density lipoprotein. So ito yung kanilang mga main lipoprotein transporters ng triglyceride. Meron tayong chylomicrons and then the VLDL. Nag-iba na lang sila kung endogenous and exogenous po ang kanyang transport. So tignan natin siya at i-discuss sa susunod na slide. But the triglycerides, its main transporter, yung tinatawag nating lipoproteins, these are mainly your chylomicrons and the VLDL. Next, the metabolism involves the releasing the fatty acids to the cells for energy, then recycling the glycerol into triglyceride. Next, the lipase, lipoprotein lipase, epinephrine, and cortisol break down your triglycerides. So ito lang po, mga additional information lang natin yan kay triglyceride and fatty acid. Let us now proceed with your cholesterol. Cholesterol is unsaturated steroid alcohol, exists in esterified form where a fatty acid forms an ester bond at carbon 3 and the free unesterified form. Next, the precursor for synthesis for bile salts, steroid hormones, and vitamin D. So, paulit-ulit po tayo dito. Pag sinabing cholesterol, this is the precursor for the synthesis of your bile acids, which is used for the emulsification of the fats in your intestine. Yung mga steroid hormones natin, yung mga progesterone, estrogen, yan. Mga steroid hormones po natin yan. And the vitamin D. Next, the LDL or the low density lipoprotein is the primary carrier of your cholesterol. So pag kay triglyceride, ang main lipoprotein na transporters niya, si chylomicrons, and then si very low density lipoprotein, nag-iba lang kung endogenous at exogenous. Kay cholesterol, ang main lipoprotein na carrier niya is si LDL. Pakitandaan po. Next, phospholipid. Let us proceed with your phospholipid. It is composed of one glycerol molecule and 2 fatty acid molecules attached via ester bond. So ano yung kapareho ni phospholipid kapag pinag-uusapan natin yung kanyang chemical structure? Same po sila ni triglyceride, phospholipid and triglyceride. Similar sila ng kanilang chemical structure. They have one glycerol molecule and then two fatty acid nga lang kay phospholipid, pero kay triglyceride sa kanya, 3 fatty acid. Okay? So this is the classification of your lipoproteins. Again, your lipoproteins are molecules that combine water-insoluble dietary lipids and water-soluble proteins so that lipids can be transported throughout the body. So micelles are spherical and have an inner core of your neutral fat. So being your lipids as hydrophobic, okay, they have the hydrophobic property, they needed this following proteins for them to be transported into the different organs of our body. Okay, so kailangan magbabound sila doon sa mga tinatawag nating proteins so that they can be transported. So yung number 1 natin doon is si chylomicrons. These are the largest lipoproteins and have the lowest density. Again, siya yung pinakamalaki, pero siya yung may mababang density. They are formed in the intestines. Again, they are formed in the intestines and transport triglycerides after a meal, giving a serum turbid appearance. Because of their low density, chylomicrons will float on the top and form a creamy layer when plasma is stored overnight. So, ibig sabihin, malaki siya pero magaan. Okay? So, pagka iniwan mo yung tinatawag nating samples mo on a test tube or your blood collecting tubes, it will float as a creamy layer doon sa plasma. Kasi nga po, mababa yung kanyang density, masyado siyang magaan. So the chylomicrons are composed of 86% triglyceride, 5% cholesterol, and 7% phospholipid, and 2% of about apolipoprotein. Okay, in a normal lipid metabolism, chylomicrons enter the circulation and are metabolized to remnants particles for uptake and further modification of the liver. So next, let us proceed with your very low density lipoprotein. It carries the endogenous triglycerides. Okay, very low density lipoprotein, endogenous triglyceride. So kung ka-endogenous po si very low density, si chylomicrons exogenous. Okay, triglycerides synthesized in the liver. VLDL molecules are composed of 55% triglycerides, 19% cholesterol, 18% phospholipid, and 8% of about your apolipoprotein. Okay, so VLDL, in a normal lipid metabolism, your VLDL are secreted into the blood by the liver for metabolism in your peripheral tissues. Kasi nga po, they carry your triglyceride endogenously. Okay, so VLDLs are secreted into the blood by the liver for metabolism in peripheral tissues. Okay, so skip na muna natin tong si intermediate density lipoprotein. Proceed tayo kay low density lipoprotein. Again, ano ba sabi natin kay low density lipoprotein? It is your body's major cholesterol carrier. Okay? Primary carrier of your cholesterol. And transports a large amount of endogenous cholesterol. So wag pong malilito pag nakita na endogenous, iniisip nyo kaagad si very low density lipoprotein. Nagkakatalo po tayo doon kung ang tinatransport ba nilang type of your fat is cholesterol or triglyceride. Again, ulitin lang natin kapag triglyceride, ang involved doon chylomicron at VLDL. Pero pag sinabing cholesterol, low density lipoprotein po yun. And it transports a large amount of your endogenous cholesterol known as your bad cholesterol. LDL is easily taken up by the cells. So elevated levels are associated with increased risk of your atherosclerosis. Okay? So for the clinical significance of your different types of lipoproteins, the abnormal lipid metabolism can be due to general genetic defects or it can be acquired. Abnormal lipid metabolism is associated with risk of your coronary heart disease and other disorders. So ito yung mga reference range natin based on our book. So hindi naman po sila applicable all throughout sa mga different clinics natin and hospitals, and depending pa rin po yan sa country. But ito lang yung sinusunod nating reference range according sa reference book natin na si Shula. So for the total cholesterol, dapat ang desirable lang dyan is less than 200 milligrams per deciliter. borderline high na si 200 to 239. So, ang total cholesterol mo is mataas na kapag nag-exceed ka na ng 240. Next naman, for the HDL, pag greater than 60, yun na yung protective against heart disease. Pagka 40 to 59, the higher, the better. And then the major risk factor for heart disease is less than 40. So next, proceed na po tayo kay LDL cholesterol reference range. Ang optimal dapat natin is less than 100 na LDL cholesterol. It is measured via milligrams per deciliter. Near optimal na tayo sa 100 to 129. The borderline high is about 100 to 159 milligrams per deciliter. Mataas na ang iyong LDL cholesterol kapag ka umabot ka ng 160 to 189 milligrams per deciliter. Kapag ka naman nag-exceed ka na po ng 190, it is considered very high. So we're done with the additional information in regards to our previous discussion. So now let us proceed with your main topic for your modules 15 to 17, which is about the nucleic acids, nucleotides. So si nucleic acid, okay, meron po tayong tinatawag na nucleotide, which are the monomers na pag pinagsama-sama natin, pwede po tayo makabuo ng polymer. And then that polymer is what we call your nucleic acid. So itong nucleotide mo na to, pag pinagsama-sama mo yan, naglagay ka pa ng another nucleotide, polymer na po siya. So that polymer is what we call your nucleic acids. So DNA molecules contain several million nucleotides, while RNA molecules have only a few thousand. So pag DNA, mas madami po yung nucleotides natin compared kay RNA, kasi si RNA single-stranded lang siya. Si DNA, double-stranded yan. So mas marami talaga siyang nucleotides. DNA is contained in the chromosomes of the nucleus, each chromosome having a different type of your DNA, and humans have 46 chromosomes, 23 pairs, each made up of many genes. And a gene is the portion of the DNA molecule responsible for the synthesis of a single protein. So, ang sabi po nila, according to the new studies, hindi daw lahat ng DNA they can code for gene. Hindi lahat ng DNA pwedeng magkaroon ng segment of a gene. Okay? So, for the central dogma of life, ito lang yung kanyang template and then the product. So, pag sinabi natin central dogma of life, it is the flow of the genetic information. So, from the DNA, pagka nagkaroon ka ng DNA replication, DNA as a template, the product will be DNA. So, DNA, makakagawa ka ng another DNA. Parang ganon. Pagka naman transcription naman, the template mo is from the DNA, it will be converted to RNA. So, the final product will be what we call RNA. Next, for the translation from the mRNA, okay, it will be converted to your final product as what we call the protein. Take note lang po that product of transcription is RNA. mRNA is not the only RNA produced during transcription. May mga nabanggit na tayo na different types of RNA that can be produced in the transcription, but mRNA lang yung pwede nating gamiting template na pwedeng i-convert to protein. Okay? Kasi yung mga ibang RNA natin, meron silang iba't ibang specific roles, and yung mRNA natin, ito yung kanyang specific role. So, but the template for your translation should be mRNA. So, kung meron tayong tinatawag na nucleotides, meron din naman tayong tinatawag na nucleosides. To convert the base into a nucleoside, you can add purine and then the sugar to the base. The linkage is an N-glycoside. So, pagka sa nucleotides, meron tayong three major components. We have the phosphate group, we have the sugar group, and then your nitrogenous bases. But for your nucleosides, tatanggalin mo lang si phosphate group, pwede na natin siyang tawaging nucleosides. So, ang matitira doon, si nitrogenous bases mo, and then yung inyong sugar. So, for purines, the connection between C1 of sugar ribose and N9 of purines. For pyrimidines, the connection is between N1 and then the C1. Okay, tandaan po, for the purines, N9 to C1 glycosidic bond. Kasi si glycosidic bond, ito yung term na ginagamit natin sa connection of a sugar to another functional group. Hindi lang sugar to sugar. Okay, pwede din po si sugar to nitrogenous bases. Ganun po. Yun yung tawag sa connection nila. Dito siya banda. So, the addition of pentose sugar to a base produces a nucleoside. So from adenine, magiging adenosine na siya. Si guanine, magiging guanosine. Si cytosine, naging cytidine. Si thymine, naging thymidine. Si uracil, naging uridine. Okay, again, the addition of your pentose sugar to a base, it produces what we call nucleoside. And then most of the nucleosides for purine ends with your sine. Yun yung kanyang palatandaan. Pagka naman nucleosides siya with pyrimidine, nagiging DINE na, hindi na sine. Si sine pag purine lang. Si pyrimidine, nagiging dine. Okay? And then if you attach a deoxyribose, the deoxyadenosine, deoxyguanosine, deoxycytidine, and so on. Okay? So, pag tinanggal natin yung deoxy, magiging ribose lang siya. H lang siya, wala yung O. So, next, the nucleotides naman, kung nabanggit na natin si nucleosides, let us move forward to your nucleotides. To convert the nucleoside to a nucleotide, yun na nga po yung sinasabi natin, mag-add ka ng phosphate. Kasi sa nucleosides, tinanggal mo si phosphate group. Pagka naman gusto mo na siyang i-convert to a nucleotide, mag-a-add ka lang ng phosphate. Okay? Nucleotides can have 1 or 2 or 3 phosphates. So, kung ganito siya, nung nucleoside siya, pag dinagdagan mo na siya ng phosphate, hindi po phosphate group ha, phosphate lang, eto siya. Okay? Pwede pong magkaroon ng 1, ng 2, or 3 na phosphate. Ito na yung tawag sa kanya. So, tingnan natin sa illustration kung paano yung naging mechanism nung nag-add na ng phosphate from a nucleoside, naging nucleotide na siya. So, tingnan natin, meron kang adenine dito and then meron kang ribose sugar dito. Okay? So, nung dinagdagan po natin ng phosphate, naging adenosine 5 monophosphate na, isa pa lang. Ito siya, ito, tsaka to. So, nung dinagdagan natin ng isa pang phosphate dito, naging adenosine 5 diphosphate. Dalawa na yung phosphate mo. Ito na, tsaka to. Next naman, si adenosine 5 triphosphate. Meron kang 1, 2, 3 na phosphate. Meron kang adenosine dito, adenine tsaka ribose. Ito na. Adenosine triphosphate na yan. Okay? So, si adenosine triphosphate, si ATP, the function of your ATP is it is your energy currency of the cell. When you cleave phosphate, 7 calories can be generated per each bond, where ATP is converted to ADP. Ibig sabihin, may na-release na energy. So, from ATP, adenosine 5 triphosphate, naging adenosine diphosphate. Ibig sabihin, may na-release na energy doon sa action na yun. So next, we have yung structure po ng tinatawag natin na DNA inside your chromosomes. So si DNA being double helical in the structure. Okay? Kapag nabalibutan siya ng mga tinatawag natin na proteins, yung histones, nagiging tinatawag natin siyang nucleosome core particle. Pero pagka nagsama-sama na siya dito, yung parang itsurang macarons, ang tawag na dyan, mga chromatin fibers na po natin. Or chromatid, pwede naman. Chromatin fibers na yan, mga yan. Okay? So, yun, kapag condensed na po siya, pwede na po silang magsama-sama and then it is heavily condensed and packed para magkasya sila dun sa chromosomes natin. So, recall lang po ulit ng structure ng ating nucleotides, particularly yung ating DNA and RNA. We have yung pinaka-ribose sugar muna, 1, 2, diba? 1, 2. Doon sa 2, doon magkakaalaman if it is deoxyribose or ribose lang. Pag ribose, may oxygen. Po siya, pag deoxyribose, walang oxygen. Kaya nga deoxyribose. Wala yung oxygen molecule or atom. Sorry. Next, 1, 2, 3, 4, and then 5. 5 carbon atom siya. Pentose. Pentose, your DNA is deoxyribose is a monosaccharide. So, 1, 2, 3, 4, 5. Dito nakakabit yung tinatawag nating phosphate group. And then dito naman sa nitrogenous bases natin nakakonect sa 1 carbon atom natin. And then tinatawag natin na N-glycosidic bond. So, for the nitrogenous bases, we have 2 types, the pyrimidines and the purines. Sa pyrimidines, may tinatawag tayong cytosine. Thymine pag DNA, uracil naman pag RNA. So sa purines, meron tayong adenine and then the guanine. So basically, ito lang yung three major components na kabisado na natin. Nagkaiba-iba na lang po sila depending on the nitrogenous bases, if it is pyrimidines or purines, and kapag ka doon sa ating sugar, if it is ribose or deoxyribose. Pero nonetheless, it is a pentose sugar kasi isa siyang 5 carbon sugar. 1, 2, 3, 4, 5. Lahat po yung mga carbons. So, ayan. Tawag dito, diester bond. Tawag dito, glycosidic bond. Yung nagko-connect between your functional groups. So, for our DNA, meron tayong tinatawag na DNA replication. So, kung nabanggit na natin yung pinaka-structure kanina, meron tayong dadaanan lang na mabilis na tinatawag nating replication. So, ang principle po ng ating DNA replication is that mag-a-unwind yung ating DNA. Wait lang. Ang DNA kasi natin, it is a template strand. It is semi-conservative, it is anti-parallel to one another, and then semi-conservative in a way na itong template strand, yung old strand mo, pag napunta siya dito at naka-create siya ng bagong strand, yung luma, mapupunta siya dun sa panibago. Parang ganun. So, but before po sila na makapag-replicate at makapunta doon at makagawa ng another daughter or yung new template strand, maghihiwalay po muna dapat sila. Okay, so mechanism niya is maghihiwalay, magkikreate ng replication bubble. Okay, so sa paghihiwalay na yun, pwede pong makabuo ng daughter cell or yung tinatawag nating daughter template. So, hindi madali yung process ng paghihiwalay ng ating DNA templates kasi medyo tedious siya. Pero madami po tayong mga enzymes and proteins that will help us para makapag-generate tayo ng isang tinatawag natin na process on what we call the DNA replication. So again, balikan lang natin na mabilis. Ang ating replication dapat po 5 to 3. Okay, so napansin ko may mali dito. Ito mali ito. 5 to 3 yan. Replication occurs in only one direction on the template strand. Dapat 5 to 3. Ayan po, 5 to 3. Evident naman. 5 to 3 sa leading strand. But in the case of your lagging strand, hindi ganun yung nangyayari. Kasi nga, semi-conservative and then anti-parallel sila. So, 3 to 5 yun nandito. So, meron tayong mga tinatawag na enzymes such as your RNA primers na kung saan tumutulong siyang magkabit ng mga RNA. Okay, pagka nagkaroon na ng mga maliliit na fragments ng RNA dito, tatanggalin niya ng mga DNA polymerase niyo, papalitan na po siya ng DNA. Okay? Pero yung unwinding, nung tinatawag natin na helicase, yung enzyme natin na helicase, siya yung tuloy-tuloy na mag-unwind dito hanggang sa magbukas na po siya doon at makagawa ng another daughter template. So yun lang, meron lang tayong DNA leading strand kung saan tuloy-tuloy lang, continuous naman yung paggawa. Sa lagging strand, lagging siya kaya medyo log kasi nga hindi po pwede na yung 3 to 5. However, meron tayong mga tinatawag na RNA primers na kung saan siya yung naglalagay ng mga RNA and then chuchugiin yan ng mga DNA polymerase natin para palitan po sila ng DNA. So yung pinaka-fragment na yan, ang tawag natin dyan, Okazaki fragment. So yung enzyme naman yung tumutulong sa atin para buksan yung tinatawag natin na template strand, yun yung mga helicase natin. And then yung enzyme naman na tumutulong sa ating eukaryotic cells para hindi sila mag-coil pagdating dun sa dulo sa kaka-unwind ni helicase, yun yung tinatawag natin na topoisomerases. So the identity of the bases on the template strand determines the order of the bases on the new strand. A must pair, yung tinatawag natin na Chargaff's rule, dapat may base pairing po talaga yung mga nitrogenous base pairs natin. Dapat si AT and CG. Si adenine, si thymine, si cytosine, and then si guanine. Okay, in the case of the DNA, pag RNA yung pag-uusapan, uracil yan. So a new phosphodiester bond is formed between 5-phosphate of the nucleoside triphosphate and the 3-OH group of the new DNA strand. So again, yung replication natin, it occurs only in one direction on the template strand, dapat 5 end to 3 prime end. The new strand is either a leading strand, which is growing continuously, and then the lagging strand growing in small fragments, or what we call the Okazaki fragments. So now, let us proceed with your RNA structure. Sorry, hindi ko na-edit to. Sorry, module 17 na ata tayo kasi kinapi-paste ko dun sa other PowerPoint natin. Okay? So your RNAs, or what we call the ribonucleic acids, meron tayong at least 4 types of the RNAs, both for the eukaryotic and the prokaryotic. But for this discussion, magpo-focus lang po tayo sa eukaryotic RNA. And then these are the differences of your RNA from your DNA. So in size, your RNA is considerably smaller than your DNA. And then for the sugar, it contains ribose sugar only instead of the deoxyribose. Okay, as evident naman doon sa ating second carbon, okay, OH and then the H. Next, for the nucleotide, it contains uracil. Si thymine kasi, meron po yan sa DNA, but for your RNA, ang nucleotide natin is for the nitrogenous base, meron tayong adenine, uracil, cytosine, and then the guanine. And then the form, it usually exists in a single strand form compared to your DNA, which is double-stranded po ang ating mga DNA. So RNA, again, nucleic acid siya. Kasi meron siya ng 3 major components. Okay, so basically, it is a nucleic acid. But it has a different sugar. And then meron lang nagkaroon ng slight differences doon sa ating nitrogenous bases. But all in all, nucleic acid pa rin naman si RNA. So going back, RNA molecules have extensive base pairing, which produces secondary and tertiary structures that are important for RNA function. So si adenine with uridine, it requires 2 hydrogen bonds, and then the guanine and the cytosine, it requires 3 hydrogen bonds. So i-discuss na po natin. Okay, daanan na lang natin yun. Hindi na po tayo mag-focus kay prokaryotic cell RNA structure. Mag-focus na lang tayo doon sa ating major classes of RNA for your eukaryotic cell. So we have 3 functionally distinct classes of RNA that are produced in the prokaryotes, pero kay eukaryotes 4. Again, kay prokaryotes 3 lang sila. Tandaan lang natin na 3, pero hindi na natin sila i-discuss isa-isa. Pero kay eukaryotic cell, merong 4. So what are these 4? But I only included 3. Bakit ganun? So for the 4 major classes, we have your tRNA as the smallest. Among all the 4 RNAs for your eukaryotic cell, ang pinakamaliit si tRNA. So ang mga major importance niya and the basic functions of your tRNA, magbasa muna tayo ng description. We have 74 to 95 nucleotide residues, 1 specific tRNA type for each of the amino acid, and then it contains unusual bases, example yung mga pseudouracil. Extensive intrachain base pairing. It serves as a adaptor molecule. It carries the specific amino acid covalently attached to its 3' end to the site of the protein synthesis. It facilitates the incorporation of amino acids into newly synthesized proteins in a template-dependent manner. So, yun yung kanyang pinaka-major function. It facilitates incorporation of amino acids into the newly synthesized proteins in a template-dependent manner. Next, yung mRNA natin, your messenger RNA, it is the most heterogeneous type in terms of the size. Kasi meron siyang 500 to 6,000 nucleotides. And then, ang kanyang pinaka-major function is that it carries genetic information from the DNA to cytosol to ribosomes. And siya yung pinaka-template natin sa protein synthesis. Again, babalikan natin yung slide natin doon sa dulong-dulo. Okay? Di ba po? Template natin, mRNA, messenger RNA. Sorry, let us proceed. Ayun. So, next naman si ribosomal RNA or your rRNA. It is found in association with different proteins as components of the ribosomes. Again, involved pa rin na naman siya sa inyong central dogma of life, but mainly, dito na siya involved sa site of protein synthesis. Involved pa din naman si mRNA, but mainly talaga siya yung template. Pero si ribosomal RNA, it is found in association with different proteins as components of the ribosomes for the site of your protein synthesis. Well, these are my references, and that concludes our discussion for today. So thank you and God bless.